Good afternoon, everyone. Thank you so much for tuning in to my webinar as part of this conference. My name is Jessica Ma. I'm an assistant professor at the Albert Einstein College of Medicine here in the Bronx in New York. The title of my talk is Investigating Tissue-Specific Networks of Cancer-Causing Mutations. And I'd like to thank you for tuning in, and please encourage you to submit questions as we go along. There's a question and answer box that you can use, and I'll be keeping track of those questions as they come in, and if there's time, I'll deal with them as we go along, and if not, we'll spend some time at the end going over those questions. So let me just get started with this presentation. And the starting point for my seminar is to step back and consider a fact that we're all aware of, and that's that there's a huge amount of cellular diversity that exists in the human body. So we know that the human body consists of over 200 highly specialized cell types. And when we look at these cells in terms of their morphology, in terms of turnover rates, structural properties, etc., there's clearly a lot of differences between different cells that exist in the human body. Now, what's remarkable is when we step back and consider that virtually all of these cell types share the same genome. So that the diversity that we can see, even at the resolution of the naked eye, is being driven by epigenetic and transcription factor regulatory programs. So when we think about a cell type's unique properties, these are regulated by its largely transcriptional program. So we look to the gene expression profiles that make up a specific cell type's unique properties. So well, we can also step back and think about cancer as a systems level disease. Now, I'm a bit biased coming from a systems biology department. We tend to think of cells and diseases in terms of how they affect the human body as a system. So in this day and age, it's important to acknowledge that cancer is actually a broad spectrum of diseases. But one of the unifying facts of cancer is that it's fundamentally unregulated cell growth. Okay, and when you think about the human body as a system, what we actually mean is you can think about different cell types being perturbed in the presence of this unregulated cell growth. And in the presence of these perturbations, some tissues go on to develop cancer and some tissues do not. So when we step back and think about it, tumors actually arise in a highly tissue-specific manner. And in this case, it's important to distinguish that we're talking about the primary tumor arising. But even when we consider metastases, often specific tumors also metastasize in a highly tissue-specific manner. Excuse me. And breast cancer is a good example. Okay, breast cancer often metastasizes to the brain and to the bone. But what I wanted to emphasize is that in this talk, we're purely talking about primary tumors. So a good area to think about cancer is in terms of germline mutations. Now we know that germline mutations predispose cells to cancer. And just so we're all on the same page, a germline mutation is defined as any change or variation in a gene that's occurring in a reproductive cell, so whether it be an egg or a sperm cell. And what's most important is that it's incorporated in the DNA of every single cell type in the offspring. In other words, the germline mutation is transmitted to the next generation. So based on all our work in cancer research, we know about 80 genes that have germline mutations that predispose to different types of cancer. And out of all the cancer genes that we know about, only about 20% have germline mutations, and about 10% show both somatic and germline mutations. So the majority of mutations that affect cancer or cause cancer are actually occurring in the soma, the somatic tissue. But what's really interesting to consider is that germline mutations are present in every single cell type of the body. So one good example to think about is germline mutations that occur in the breast cancer gene BRCA1. Okay, now we all know that BRCA1 is linked to hereditary breast and ovarian cancers. And in normal healthy tissue, BRCA1 functions as a tumor suppressor gene. It's actually critically involved in repairing DNA double-stranded breaks, and it's also involved in mismatch repair. Now, BRCA1 is well known in terms of cancer research because mutations in BRCA1 account for 80 to 90% of hereditary breast and ovarian cancers. And it's actually also involved in over 50% of inherited breast cancers. Now, what's really interesting to consider is that you've got a patient who has a germline mutation in BRCA1, and they're predisposed to develop cancer in the breast and in the ovaries. But when you think about the other tissues, these are unaffected, and from our perspective, you could consider them to be cancer resistant, whereas breast and ovarian tissue are cancer susceptible. And so what is it that distinguishes the breast and ovarian and other tissues from being affected by the germline mutation? If you think of perturbing the system, every single tissue in the system is perturbed due to the presence of the BRCA1 mutation. However, this perturbation results in the development of a tumor only in the breast and ovarian tissues. And when we consider even more what's going on with BRCA1, it's important to note that BRCA1 is highly expressed in many different kinds of cell types, both at the level of transcription and also at the level of proteins. So what we want to ask ourselves to try and answer this question is, how is BRCA1 regulated in a normal setting, in a cancer-free setting, in different tissues? Okay, as I mentioned, we do detect high levels of BRCA1 in different tissues, and this indicates that BRCA1 is an important gene product in multiple cell types. So as we talked about before, each tissue in the human body is governed by its own tissue-specific regulatory networks. They share the same genome, and the differences that we see from tissue to tissue are explained by changes in gene expression and the wider regulatory networks that control these cells' properties. So what we want to do, using BRCA1 as our example, is look in normal tissue to understand how tissue-specific differences affect a cell's ability to buffer in the event of a germline mutation occurring in BRCA1. So what we're trying to do then is to actually reconstruct and analyze tissue-specific gene networks to determine whether there are hints of local network changes that can explain the susceptibility or resistance to cancer that we observe in the susceptible tissues. Okay, the whole goal of this is to understand why some tissues are able to tolerate germline mutations better than others. And we're using genes with germline mutations as a test case because they're present in every single cell type of the human body. So just to step through the analysis that we've done, the research that we've done, this is the flowchart of our work. So first of all, and I'm going to go through each of these steps in more detail, but the goal of our work was to first of all start with a subset of genes where we know that they cause cancer and through germline mutations. We then wanted to identify which of these genes that are associated with germline mutations have tumors that are highly tissue specific. We then wanted to look at gene expression across a tissue atlas to understand how these genes were being expressed differentially between different tissues. And then what we've done is to go ahead and both understand what these different classes of genes are doing, but more importantly, construct different types of gene networks And in this case, we've used protein-protein interaction networks and transcription factor networks. And finally, we look at functionally at how these may shed insight into the buffering capability of these cancer-susceptible and cancer-resistant tissues. Okay, so first of all, what we needed to do was identify a set of cancer genes with germline mutations. And the resource that we turned to was the Cancer Gene Census. Now, this is an amazing catalog of genes that genes in a repository that's been hosted through the Sanger Center. These genes have all been identified as having a causal role in cancer. And the census contains all these cancer genes. They make up about 1% of all human genes. And basically there's a wealth of genetic information that's been reported from the literature that's captured in this gene census. So if you're interested in having a look, I'd highly recommend this as a resource. And from this, we've been able to extract about 80 different genes that are linked with germline mutations that cause cancer. That number has changed with regular updates from the resource. So now that we have our set of germline genes with germline mutations, it's important to step back and understand that just because a gene has a mutation doesn't mean that you'll automatically, definitely get cancer, okay? And to explain this idea further, there's a concept in genetics related to penetrance, and this is the idea in this context that not all tissues carrying the same germline mutation will develop cancer. And in fact, what we see is that some germline mutations are associated with cancers that are highly tissue-specific, like BRCA1, while others affect tissues more ubiquitously, meaning that a lot of different tissue classes are affected with the same germline mutation. So in this context, we define tissue-specific penetrance as the property of observing a large number of tumors associated with germline mutations that are occurring in genes in one or two tissue groups. And so obviously that one or two is an arbitrary definition, but the point that we want to get across is that this is a highly tissue-restricted subset, okay? And to get that information and to actually assess tissue-specific penetrance, the resource that we've turned to is to use COSMIC, which stands for the Catalog of Somatic Mutations in Cancer. Now, this is also another resource that's hosted by Sanger. It contains a wide array of genetic information on cancer samples that have been collected in the literature. And this information, it's worth pointing out, pertains to both germline and somatic mutations, despite the somatic in the catalog's name. So basically with COSMIC, for every gene that's listed, you can look at the mutations as they are reported in about 40 standardized tissue categories. So you can get tissue-specific information on specific mutations that are reported in specific genes. So for instance, you can say for gene X, how many samples have this mutation that we know about in the current literature, and how many of those samples occur in a specific tissue, say tissue Y. Okay, and so just to give you a demonstration of that, we have the tissue-specific profile for P53. Now we all know P53 is a very important gene in cancer genetics. And here what I'm showing you is the tissue percentage distribution for P53 in COSMIC. Okay, and it's really hard to see the labels on the x-axis. I apologize for that, but basically these are the different tissue categories. So for instance, and I'm afraid you're just going to have to believe me, but one of these bars corresponds to lung, breast, ovary, brain, CNS, and the height of the bar reflects the percentage of samples out of all the samples that have P53 mutations that are specific or reported in that particular tissue class. So the point of this profile, or this graph is to show you that for P53, it's actually quite scattered in terms of the tissues where mutations in P53 have been reported. And that is no surprise in terms of understanding what P53 does in cancer. Now, it may be hard to see, but hopefully you can see also the red line that is going across that chart. And basically that represents the tissue mean. So in cosmic, different tissue classes have different numbers, absolute numbers represented in the catalog. And this obviously reflects the fact that some tissues are easier to profile than others. It's easier to get a biopsy from the breast or the lung rather than, an eye or the brain, for instance. And so in doing our calculation and trying to figure out which genes were tissue specific, we didn't want to be biased by the sampling that was present in cosmic. So what we've done to determine whether a gene is tissue specific in terms of penetrance is we've taken those genes that have a tissue percentage that's greater than the tissue specific mean in one or two different tissue groups. And so to help elucidate that concept, let me show you the tissue specific penetrance profile for BRCA1. Okay, so in this case, what we've got now is the same kind of profile. We're looking at the same tissue categories that are represented in cosmic, and you can see automatically that BRCA1 is highly tissue specific for the breast, which is to the left, and the ovary, which is sort of in the center of the graph. You can see those really high bars compared to all the other tissue specific categories. So another thing to note is that the next highest bar that's not labeled is actually the lung. So this may not be such a surprise. But what is interesting is that you can see the tissue mean for the lung is actually generally quite high. And I think that reflects the fact that there is a large percentage of tumors in the cosmic database that are a lung tissue. So what we've done, just to repeat, is to count the number of tissues associated with a gene where the tissue percentage is higher than the tissue mean. And that forms our definition of a highly tissue-specific penetrant gene. And so just to look at this now, what we see is that for the 70-80 genes with germline mutations, most of these germline mutations are actually associated with tissue-specific tumors. And so what we've got here in this graph is basically a histogram where we're counting the number of genes with germline mutations and the number of tissues a gene is mutated in. Okay, and so here you can see some of the typical cancer genes that are represented, such as ATM, BRCA1, BRCA2, EGFR, APC, which is associated with colorectal cancer, HRAS, P53, that's all the way at the end, at the high end of the number of tissues. And we have two genes, DDB2 and TSC2, which we didn't have data on. That's why there's 0 tissues. There's not enough data there. So basically what we've done is we've drawn a line, and I wish you guys could see my mouse pointer, but what we've done is drawn a line between 2 and 3, and we've called everything to the left of that line tissue-specific, and everything to the right ubiquitous. Okay. Now, when we think about how these genes are operating in different cell types in the human body, we have to consider the level of gene expression of these genes as well. We know that there's a germline mutation that's present in every single cell, and only a subset of those tissues are susceptible to cancer. So when you consider genes that are highly expressed in a wide range of tissues, we have to ask the question of how resistance is achieved in some tissues but not others. So to go back to our BRCA1 example, we can imagine that we know for BRCA1 that the breast and ovarian tissues are not resistant to cancer, and yet we can consider, in terms of neural cells or brain cells, the incidence of primary events of cancer is very low, okay? But yet when we consider transcript availability and levels of expression, we see high levels of expression across different cell types. So it's not a simple situation of a gene is mutated in a specific tissue and therefore the level of gene expression is decreased. It's not a dosage effect in the absence of the ability to produce this transcript that the downstream effects lead to tumorigenesis. Okay, so in order to look at gene expression in a tissue-specific manner, we've turned to a resource that's been made available by the Fantom5 Consortium that's being led at the Riken Omics Science Center in Yokohama in Japan. So the goal of the Fantom5 Consortium was to build a complete promoter map to uncover the transcriptional regulatory networks that define every human cell type. And these guys have created a really great resource that will be published and released soon. But this resource represents a tissue-specific atlas that covers over 1,000 human primary tissues and cell lines that have been collected from all over the world. And so underlying this atlas is the Heloscope CAGE technology. And basically what this is, it's a technology that allows one to capture promoter usage. And so this technology uses capped analysis of gene expression, and this is a single molecule sequencing technology. There's no amplification step involved. It's possible to start with very small amounts of RNA. And the idea is that for each transcript, this technology sequences the 5' cap of the mRNA. So in this way, the technology is quantitative. We can count the number of tags and get a surrogate measure for how expressed a gene is. But moreover, we can actually also identify where the transcription start site is occurring. And in this way, we can get a sense for how promoters are being used across the genome for different cell types. So what we've done then is to take the gene expression data from the Fantom5 resource and look at the expression of our genes with germline mutations across different tissues. And the kind of situation that we want to distinguish from are genes that are tissue-specific in their gene expression from genes that have a ubiquitous profile. And here I've taken two genes to highlight what I mean. On the left, we see the gene expression profile for ULK. ULK is a tissue-specific, it's a gene that affects tumors in a tissue-specific manner. It's responsible for a neuroblastoma. And as you can see from the graph, it has a highly tissue-specific profile, meaning that the majority of genes are actually lowly expressed, the majority of tissues, we see low expression with some outliers towards the end, okay? And for BRCA2, what we see in contrast to the ULK profile is this has a much more ubiquitous expression because we see multiple different states. There's quite a broad expression distribution, meaning that there are multiple different tissue classes where we see different levels of BRCA2 expression. So when you think about this, I think, hang on a second. All right, so what we've done then is to synthesize these two concepts related to tissue specificity in terms of penetrance and tissue specificity in terms of gene expression. And we've created a classification for the genes with germline mutations. And we've distinguished three different classes that are of interest. Now, the first class represents those genes that have a mutation causing tumors in a tissue-specific manner, and where the gene is expressed only in that tissue. Okay, so we can imagine the direct link between a gene that causes a tumor in a specific tissue, and it's also expressed, highly expressed in that particular tissue. We can also consider a second class where a gene with a mutation that causes the tumors occurs in a highly tissue-wide manner. In other words, it affects lots of different tissue classes, and it's also expressed in a lot of different tissue classes as well. And finally, our third class, which to us is the most interesting class because it represents the genes that have mutations that cause tumors in a specific tissue and are also expressed ubiquitously. So like the situation with BRCA1, where we know that the tissues that are susceptible to cancer are highly tissue specific and restricted to the breast and ovaries, and yet we see expression across a lot of different kinds of cells or cell types. So these are the ones we actually want to focus on. And out of our 70 or 80 genes, this is the distribution of counts in terms of how they fall in the different classes that we've delineated. And I'll go into more detail in a few slides' time in terms of what those genes actually are. But first of all, I just want to go into a little bit more detail in terms of how we distinguished between tissue-specific and ubiquitous gene expression. Okay, now this is an interesting problem from a bioinformatics perspective, where we want to identify tissue-specific expression via some measure of skewness. Okay, as I mentioned before, this kind of skewed profile suggests tissue-specific nature as opposed to a much broader expression. Okay, and so from a bioinformatics perspective, the way we chose to tackle this problem was to use mixture modeling techniques. Okay, so what we did was we fitted mixture models based on normal mixtures using equal variance, and this was done using the M-classed R package. And the goal here was to capture the shape of the expression distribution using one or more normally distributed components. Okay, in a perfect world, we can imagine that the number of tissue-specific states in terms of gene expression is very distinct. We may have 1, 2, or 3 different states, and we can see them in the distribution perfectly. But the issue here is that there is bleeding between different states. For some genes, we don't know how many different gene expression states make sense to describe this particular gene. So mixture modeling tools give us the flexibility of being able to anticipate or estimate an unknown number of mixtures or different components. So just to give you more detail, we use these mixture models to fit different components to the gene expression distribution. And the rule that we had was if the critical regions associated with each of these mini distributions overlapped, then the gene is classified as ubiquitous, meaning that there's a broad gene expression distribution that we're seeing. Otherwise, if the critical regions were so extreme that there was no overlap, we consider that to be a tissue-specific expression profile. Now, as you recall from basic statistics, the critical region is the region defined by usually half of the type 1 error rate. So in this case, we're using 0.05, so the critical region was defined by the 0.02 basically here. And so from our mixture model, we can get estimates of mu1, mu2, and sigma as well. So what happens when we have more than two adjacent mixtures or mixtures in the gene expression distribution? So in this case, we've extended our rule such that all critical regions between any two adjacent mixtures must be distinct for the gene to have a tissue-specific expression profile. And so this is just the requirement that all components that have the potential to overlap are extreme enough, meaning the states or the gene expression states that they correspond to are distinct enough or extreme enough such that they don't overlap. Okay, so this was the rule that we applied alongside the rule that we used to determine tissue-specific penetrance of the tumors. And as a result, these are the cancer genes that segregate into the 4 classes that we talked about. So class 1 represents the tissue-specific tumors as well as those genes for tissue-specific expression. For class 2, these are the genes that have ubiquitous tumors and ubiquitous tissue expression, gene expression. For class 3, again, these are the most interesting genes because they're tissue-specific in the tumors that are affected, but ubiquitous in the gene expression that we observe across the tissue atlas. And finally, we have this kind of strange group of genes, which we haven't really explored at all. And these are the genes that are known to be mutated in a large range of tumors, but are highly tissue-specific in terms of their gene expression. So, first of all, we wanted to ask within these different groups of genes, what kind of functional roles are enriched or shared within these classes. And so the first thing we did was to look at ubiquitously expressed genes versus tissue-specific expressed genes and try to figure out what sort of categories or roles in terms of cancer and general biological processes these guys participate in. And one of the most striking results that emerged from this analysis was that ubiquitously expressed genes are largely tumor suppressor genes. And the kind of categories we see based on gene enrichment was cell cycle, DNA repair, cell adhesion type pathways and processes. On the other hand, when we look at tissue-specific expressed genes, we see that largely these are oncogenes. And we see terms like proliferation, differentiation, migration, and survival that are significant for this group of genes. And what's also interesting is when we look at our class 3 genes, these are the genes that are tissue-specific in the tumors affected and ubiquitous in terms of gene expression. These are actually populated with tumor suppressor genes. Okay. So the next area we wanted to look at was in terms of the class 3 genes. We wanted to test out this hypothesis that tissue-specific networks are actually working to buffer the germline mutation genes in a local manner. So we can imagine that for our set of cancer-resistant tissues, we have one particular network configuration for BRCA1, and for the susceptible tissues like the breast and the ovary, there's a different local network structure that is regulating BRCA1 in a normal situation, in a normal healthy tissue, such that in the presence of a BRCA1 mutation, this network is affected differently, such that differences in the susceptible tissue make the tissue more susceptible, and differences in the resistant tissue make the tissue more resistant. So this is the hypothesis that we wanted to test in our local networks. And what we did was we used 3 different resources to construct these networks. So first of all, we looked heavily at protein-protein interaction networks. And the resource that we used was IREF index. IREF index is a resource that combines protein-protein interaction information from multiple primary databases. And it does this really well in a non-redundant fashion. So what you end up is interactions that have been reported across all the major protein-protein interaction databases. And you can access this using IFR, which is an R package. that's very easy to use. We've also used a database called TIGER, and this represents tissue-specific gene expression and protein expression, and this comes from Johns Hopkins University. Finally, we've also used information from Fantom5, where we've also used the tissue-specific levels of gene expression to highlight which genes or which protein-protein interactions are relevant to a specific tissue. Okay, so the example that I've shown here also is the networks that we've reconstructed for BRCA1. And so it's a bit hard to make out the individual gene components, but the point of this is that we have a protein-protein interaction network for the breast on the right, and the central protein here is BRCA1, so everything is centered around BRCA1. And on the left we have the protein-protein interaction network for an example of a resistant tissue, which in this case is small intestine. And basically you can see that there's some differences in the highlighting between, or the shading between different networks. So red indicates presence or absence of this protein, and there's some others in terms of the size of the nodes, in terms of levels of gene expression and how they differ from different tissues, but those are quite difficult to see in the graphics, so we won't go into that too much at all. But another way to look at this, which is better in terms of just eyeballing, a more quantitative way to look at this would be to start looking at properties of the network. So for instance, we can look at connectivity or the degree distribution associated with each of these protein-protein interaction networks. So what we've shown here is the degree distribution for BRCA1. This represents the number of protein-protein interactions for BRCA1 in each of the different tissues. And so each of the lines, the red line represents data that's been used from both TIGER and Fantom5, and then the blue and the black line represents using only one of these resources, so Fantom5 and TIGER respectively. But the point of this diagram is to show that across the different tissue classes that we're looking at, there are some differences in connectivity for BRCA1 in normal tissue. And so, but overall, we see that there is actually a lot of consistency apparent. So what I've highlighted here with these yellow bars is the ovary on the left, and on the right yellow bar corresponds to the breast network. And you can see that for these guys, there is some differences to other tissues, like stomach and intestines. And the green bars in this situation highlight the network that's been built for cancer samples that we had available. And so on the far left, we have the breast cancer sample, and on the far right, we have the ovarian cancer sample. And you can see that for these 4 tissue networks that we've highlighted, the connectivity is actually virtually identical. So there aren't any obvious differences here for these cancer and tissue-specific networks for the tissues of interest, but it could be that there are other differences in the network that we've yet to really fully explore. So what we've also done is to look at the BRCA1 protein-protein interaction network from a functional perspective. And so what we've done is we've looked at enrichment of particular pathways or biological processes of the genes that have been annotated to these protein-protein interaction networks. Now we know that BRCA1 participates in DNA damage response, DNA damage repair, so it's probably not a huge surprise that when we look at functional enrichment, we see terms relating to double-stranded break repairs and mismatch repairs. When we look at these networks, we see that breast and ovarian tissue specific networks are similar to each other, as we pointed out in the previous graph, and they're actually more similar to networks that represent the CNS and meninges. They're actually the most distinct from tissue-specific networks that come from the stomach and bone. So we're exploring some of the more we're exploring this result in more detail. And so this raises the question overall in terms of whether differences in DNA damage pathways are being used in normal tissues to determine resistance or susceptibility to cancer. And now when you think about this from just a common sense perspective, this makes a lot of sense. Different tissues in the body are subjected to different levels of DNA damage, whether it's at a basal rate, whether it's at a turnover rate that's different from one tissue to another, or whether other tissues, certain tissues are exposed to different environmental exposures or toxin exposures. It makes sense that different tissues are therefore dependent on DNA damage pathways at a different level, okay? And so from these results, we're starting to think whether these differences in DNA damage pathways are actually setting up this buffering capability of genes in the presence of a germline mutation. So what we also wanted to do was identify key components of the BRCA1 network. In terms of looking just straight at different connectivity, there wasn't an obvious difference. But what we wanted to do was look at differential expression between BRCA1 interactors between susceptible and resistant tissues. And so what we did here was we highlighted genes in the BRCA1 protein-protein interaction network that were differentially expressed between resistant and susceptible tissues, and also those that were highly connected. So we took a threshold of 10 other proteins that were reported, and what we did was we found some interesting genes that represented cyclins, kinases, genes that were involved in DNA binding. And when we looked at enrichment of functional categories in this group, they recapitulated the functional categories that we saw before when we looked at the entire BRCA1 protein-protein interaction network. And some of these examples are actually quite interesting from a cancer genetics perspective. Some of them are listed here, and we're going to be following up with them in more detail as well. So we've also looked at transcription factor regulatory networks as well, and we're using MARA, a method, or rather we're using an output from MARA, which is a method for network reconstruction that's been developed by our Fantom5 collaborators. And so here we've used the transcription factor networks that they have constructed for BRCA1, and we've looked at this network in different tissues. You can see that there is actually a lot more diversity that's observed in the network connectivity for BRCA1. And so we're starting to look at what some of those differences are. Again, in more detail, this is a result that we've only just recently got. One thing that we've also done is to extend the predictions that we've made for the cancer genes with germline mutations and extend this to the rest of the genes in the human genome. And so basically we can make predictions into what genes fall into the 4 classes that we delineated using the classifier we developed for gene expression, and also the tissue-specific penetrance classifier we developed for data in COSMIC. And as a result, you can see that these are the different numbers that fall into these different classes. And the reason why this is valuable, oh, hang on, let me just go through some of these other results. So when we look at these numbers don't really mean much, but when we look at what genes are actually falling into each of these classes, what we see in terms of functional enrichment is, again, the presence of tumor suppressors and tumor suppressor categories, or categories associated with tumor suppressor activity that are occurring in our class 3 genes. Another thing that's worth mentioning is that other significant terms are occurring in class 3 that suggest other buffering mechanisms, for instance, ubiquitination. So these predictions are putative. They haven't been experimentally validated, but the reason why they're interesting and could be of value is that they represent classes of genes to target or investigate in the case of a tissue-specific cancer, particularly if not a lot is known about the genetics of that in that specific tissue. Okay, so we'll put those lists out there soon. Okay, I'm running into question time now, so I just wanted to wrap up my presentation by going over some of the conclusions that we've been able to make so far. Now, just to remind you, the goal was to understand why resistant tissues are able to buffer a germline mutation better than susceptible tissues. We look for clues in tissue-specific networks in normal tissue to understand how regulation is occurring differently and how this might lead to susceptibility and resistance. We identified a set of class 3 genes. These were genes that had broad expression across the tissue atlas and were associated with highly tissue-specific tumors. Now, these are candidates for looking at local network changes in normal tissue to try and understand how these network changes are contributing to cancer susceptibility. For BRCA1, the evidence we're seeing is that DNA damage pathways may be the distinguishing factor between resistance and susceptible tissues. We've also identified differentially expressed and highly connected nodes in the protein-protein interaction networks for BRCA1 that may be candidates for specifically driving this distinction. And so I just wanted to end there by acknowledging some of the collaborators that we work with. We've been working with the Riken's Omics Science Center in Yokohama, specifically with Dr. Al Forrest and Dr. Mikhail Dahoon. They've been a wonderful support in terms of providing access to this resource and help along the way with this project. And finally, I'd like to thank my postdoc, Dr. Danny Carbajo-Pedroza, who has been the main driver behind all this work that I've presented today. So finally, I'd like to thank all of you for tuning in to my presentation, and I'm just going to switch over right now to take questions. So we have our first question from Ezra. Thank you, Ezra, for submitting this question. And Ezra says, what is the percentage of epigenetic markers such as histone modification or methylation in tissue-specific penetrance for the BRCA1 gene? Did you check both of them on BRCA1? That's a fantastic question. Unfortunately, with the resources available, we didn't have the ability to look at epigenetic regulation. Certainly that would be something of interest due to the epigenetic nature of tissue-specific differences. So I can't answer your question because we didn't check them for BRCA1, but that's something that would have been really interesting if we could. Are there any more questions from the audience? If that's the case, then I'd like to wrap up by thanking everyone for your participation. And I'd also like to thank the organizers of the BioConference Live. It's been really great to be able to share my research with you today. So thank you very much.